All right, let's jump right in to the CHIP Landmark Ideas Seminar. Today, we're going to hear about genomic approaches to cancer precision medicine by Todd Golub, director of the Broad Institute of MIT and Harvard. I'm Ken Mandel. I direct the Computational Health Informatics program here at Boston Children's Hospital. And our program was founded in 1994. It's a multidisciplinary applied research and education program. You can learn more about it at www.chip.org. And this is a talk in the Landmark Ideas series, which is an event series we lead, featuring thought leaders across healthcare, informatics, technology, science, and more. We're going to have an introduction to Todd. Todd will give his presentation. You can put your questions in the Zoom Q&A as we go, and then we'll have some discussion and closing remarks. Todd is the director of the Broad Institute of MIT and Harvard. He's a founding core member and director of the Gerstner Center for Cancer Diagnostics at the Broad Institute. He's also a member of the faculty of the Dana-Farber Cancer Institute and Harvard Medical School. I'll just briefly recall, I had already known Todd for many years, but this really let me know that he was an incredibly creative scientist. This is 2002, and he took microarray expression technologies, which were pretty new at the time, and used them with a clustering analysis to actually be able to distinguish different cancer types from each other. Here, acute lymphocytic leukemia from other leukemias with translocations, and he was able to do it with gene expression. This was also impressive was Todd's cooking ability. As I had never heard of salmon served on cedar planks. Todd introduced me to this as well as some pretty amazing blintzes. So I wanted to just give you a flavor that he is a Renaissance man and has been at the leading edge of cancer genomics, really from the beginning of the field. If you're interested in social media, here's Broad Institute, CHIP, and again, put your questions into the Zoom Q&A as we go, and I'm going to turn it over to Todd.
Great, thanks, Ken, for the invitation. I have to say before we get started, I don't think I would have made it through pediatric residency without your humor running alongside. So it's great to reconnect today. So let me share my screen. Okay. Quickly, a few disclosures that are listed here: consulting and research funding. Before sort of launching into the heart of what I want to talk about today, we're around the 20th anniversary of the sequencing of the human genome, and it depends when you count the initial draft or the kind of final draft, around 20 years. And it's worth asking, like, what don't we know yet? And it's clear we don't know a whole lot, particularly about how the non-coding genome works. But at least, presumably, we know what all the protein coding genes are. Maybe. And that was the point that John Prenzner, pediatrician and pediatric oncologist in the lab, asked this question. Maybe there are non-canonical proteins, in particular small ones, that are lurking in the genome, hidden in intergenic regions, in upstream or downstream regulatory regions of the genome, in alternative reading frames, and so on. And maybe some of those really do exist and are functionally important. And so he took on a computational effort based on ribosome profiling, which, as you may know, is a way of inferring proteins that are expressed in a cell based on the proximity of mRNAs to the ribosome. So if you capture an mRNA in a ribosome, you can infer it's probably being translated. This is kind of a noisy assay. So the computational predictions that come from that, I think one has to take with a grain of salt. And so what John asked was, well, could we take predictions of a couple thousand of these non-canonical open reading frames, or ORFs, that were computationally predicted based on ribosomal profiling, and actually do experiments in the lab with them, not choosing your favorite one, but doing something systematically. And so he collected the top, turned out to be 553 non-canonical open reading frames, cloned them into expression vectors, and did experiments with them. The details don't matter, but he's able to show that the majority of these non-canonical hidden proteins encoded by the genome could in fact be translated into proteins. And you could detect them as transgenes. You could also detect them by mass spectrometry if you look for them, that is looking at endogenous proteins. So these computational predictions, not all of them, but a good number of them actually do encode proteins. He then asked, well, is there any reason to believe that they're actually functionally important? Because maybe there's just kind of serendipitous translation of open reading frames, but they don't actually have functional consequences. And so one way to approach that is to knock out those genes in a panel of, this was 8 cancer cell lines, and ask whether any of these non-canonical ORFs are actually required for the survival of cancer cells. And he found that about 10% of these previously unrecognized proteins are encoded, are required for survival. And to put that into context, if you do the same experiment with the known about 20,000 canonical proteins and knock them out in the same panel of 8 cell lines, the rate of positivity is about 17%. So yes, it's lower for these previously unrecognized proteins, but not by like orders of magnitude. It's within a couple fold. So I think that's all I want to say about this, except to say that, even for what would seem like the easy aspects of the human genome, there's still an enormous amount to discover, some of which may be biologically important, developmentally important, if not representing interesting therapeutic targets for cancer. Now, I want to spend most of my time talking about what most people now call cancer precision medicine, which is the idea that one would take a tumor sample, could be a blood sample these days, but some kind of a sample of a tumor, and characterize it molecularly. And so here I use the word tumor genome kind of loosely, not just to mean DNA, but systematic, comprehensive molecular characterization. And from that characterization, we hope to infer the appropriate targeted therapy for that particular tumor in that particular patient, with the hopes that that would result in not just a transient, slight clinical response, but a real durable response and ideally cure. So this is a very attractive paradigm. It always works in PowerPoint. It sometimes works in the clinic. I think it's fair to say that the majority of cancer patients in 2022 still don't benefit from this paradigm. I think there are 2 ways to think about that. One is that, well, the paradigm's wrong, and you should come up with some other idea for how to tackle cancer therapy. Or the alternative hypothesis is that there's just a lot more we need to learn about how to make this effective for the majority of cancer patients. And I strongly believe it's the latter, and that's what our lab has been focusing on. What do we need to figure out in order to make this a more generalizable approach? So I'm going to say a little bit about that. Of course, there are at the highest level, 2 ways that you could try to learn these rules. Again, with the goal being predicting therapeutic response, predict response of a targeted therapy given the molecular characteristics of the tumor. You can learn from patients, or you can learn from experiments in the lab. Learning from patients, very attractive. because they're patients, they're not test tubes. But there are also limitations from learning from the clinic because, of course, any individual patient is only subjected to a limited number of treatments, perturbations in people, essentially, from which you can learn. So any individual patient may only see one treatment regimen, or maybe at most a few, whereas the experiment you'd really like to do is systematically subject every patient to every possible treatment and learn from that data set, but for obvious reasons, that's not possible. In addition, you're limited only to those drugs that actually exist in a form that can be given to patients. So that gives you only a sliver of the types of perturbations that are possible to do in patients. On the other hand, what you can learn from experiments in the lab where you really can do systematic perturbations of a tumor of a given genotype, treat it with all possible drugs, or even if there's not a drug yet, treat with genetic perturbations that we can do now with CRISPR, for example. That being systematic is really powerful. But of course, these are cell lines growing in a petri dish that don't have the full physiologic relevance. My own view of this is that, rather than engaging in some philosophical argument over which of these is better or more powerful, I think the obvious answer is that you really need to do both, and you need to think about them together. And so I'm going to say a little bit about what's happening at the Broad in each of these areas. First, on the clinical side, and this is really the work of Nick Wagley, a colleague at the Dana-Farber and the Broad, that says, okay, let's maximize what we can learn from the patient experience, patients treated with cancer. Before we went live here, Ken was extolling the virtues of the oncology community and how we collect all these data from cancer patients across the country, whereas that's not happening so much in other diseases. And I think that's only partially true to the extent that it's only the minority of patients, maybe 5% of cancer patients are enrolled in clinical trials. And most patients are not even treated in major cancer centers. They're treated out in community. And we don't learn anything from that community experience, even though patients themselves are saying, hey, I want to contribute, I just don't have an opportunity to do so, to contribute to research. And so the question here was, could one use social media in particular as a way to directly engage patients, not through institutions, but to directly engage patients as partners in research. And this led to what an initiative that we're calling Count Me In, which is the idea that we want to make it possible for any patient anywhere to raise their hand and say, count me in. I want to contribute my genomic information and my clinical information to research, focusing on cancer in this case. Obviously, this is generalizable and is being generalized elsewhere in the Boston HMS ecosystem to other areas, but I'm going to focus on what's being done here in cancer. The first flagship project here being in metastatic breast cancer, where paradoxically, while we know a lot about the genomics of primary tumors, breast cancer, we know very little about the genetics of metastatic breast cancer. Several other Count Me In projects have subsequently launched, including some in pediatric oncology and other adult tumor types. And this has resulted now with the accrual of more than 10,000 patients across the US and Canada, representing all 50 states, which is pretty cool as a mechanism to complement what's happening through major cancer centers like the Dana-Farber, Mass General, MD Anderson, Sloan Kettering, and so on. I think what's really fascinating is illustrated on this slide. Here, looking at the more than 2,500 patients where we had detailed information. Only 6% of those 2,500 patients got their care at a cancer center that tends to engage in research. the vast majority we're seeing out in the community with over 1,000 institutions, in some cases the institution being a private practice oncology clinic in the community, contributing in some cases just a single patient. And so I think this has the potential to scale even further, to engage patients across the country in an exciting way. And we're seeing, I'm not going to go into detail here, but starting to see that particularly for rare cancers, one can get to the scale that's needed for discovery in this kind of direct-to-patient way that you really can't in traditional ways. I think this also has the potential to increase the diversity of patient enrollment compared to what we see just happening to come through the door in major cancer centers. Now, I want to turn next to the other side of this coin, which is learning from the laboratory and models of cancer. And here, one of the flagship projects of the cancer program at the Broad is a project that we're calling the Cancer Dependency Map, or DEP map. So the concept here is that we have models, cell lines derived from patient tumors that can now be comprehensively characterized, genome, proteome, transcriptome, so on. And because they're scalable models, we can subject them to systematic perturbation. For example, with genome-wide CRISPR knockout screens, or large-scale small molecule screens, drug screens. And from this systematic perturbation of genetically characterized tumors, develop a map of cancer dependencies. And this is a large effort. Scientific leader is Paquita Vasquez at the Broad, with computational leadership from James McFarland, who leads our cancer data science team, and Bill Hahn on the faculty side, also leading this effort with previous leadership by Jesse Bohm. So again, the idea is to say, all right, if you've got this data set of hundreds of cancer cell lines, each of which is characterized with more than 100,000 features, mutation, copy number, RNA, protein, metabolite, so on, and we have a dependency profile, that is, we know across the entire genome which genes, when knocked out, are required for survival of that particular cell model, one can then develop predictive models of this gene essentiality, which can point to therapeutic targets. I'm not going to say really anything about the computational approaches that we're using here, in part because I'm not a computationalist myself, but I would also say that so far, I think what we're learning is that the most straightforward, in some cases simplest models, tend to have the best performance, and have not yet seen evidence, although very much open to it, that more advanced ML-based methods are pulling out really robust relationships that were previously unrecognized. It may be that we're not yet at the scale of data, or not using all methods that are available, but that's kind of been the Gestalt observation so far. So I'm going to give one example of the kinds of things that we're discovering here. The work of Dan Bondison, a postdoc in the lab, who is interested in ovarian cancer. And Dan asked the question of this DEPMAP data set that, again, is about 800 cell lines with genome-wide CRISPR knockout, all 20,000 genes in pooled format across each of the 20,000, each of the 800 cell lines, and asking, are there any genes that are selectively required for the survival of ovarian cancer cells compared to other tumor types? Because if you knock out a gene that's required for the survival of all cancer cells, all 800, let's say, yeah, it's possible that that's going to be a target for the world's best cancer drug, but it's much, much more likely that that's just going to be a universally toxic, common essential gene, that if you made a drug that phenocopied that, you'd just get horrible toxicity and would not be tolerated. XPR1, though, with selective ovarian cancer dependency was interesting, and it turns out that XPR1 is a phosphate exporter. It's the only phosphate exporter in mammalian cells that at least has been characterized. And what's interesting is that then when we looked at the predictive models and asked which features are most predictive in the top models of dependency on XPR1, it's the expression of a gene called SLC34A2. And a reminder, XPR1 is a phosphate exporter, and it turns out that SLC34A2 is a phosphate importer. So that is, if you express high levels of the importer and then you inhibit the exporter, the model suggests that you have an intracellular accumulation of phosphate that results in a toxic accumulation and ovarian cell death. I'm not going to go through the experimental validation of this. We're able to validate this in vitro and in vivo, and to show that indeed you do have intracellular phosphate accumulation in these XPR1 knockout SLC34A2 high ovarian cancers, leading to the following model, which is interesting to the extent that, to my knowledge, phosphate homeostasis had not been previously considered as a thing with respect to cancer therapeutic strategy. So under normal conditions, as shown on the left, you have this normal balance of phosphate uptake through the SLC34A2 importer, balanced by phosphate efflux, the XPR1 exporter. In the case of ovarian cancers, excuse me, those cancers are driven by an ovarian cancer lineage-specific transcription factor called PAX8. And it turns out that SLC34A2 is a downstream target of PAX8. And so in these ovarian cancers that require PAX8 for survival, that drives super high levels of SLC34A2, the importer. And so there's a compensatory upregulation of XPR1 in order to keep the cell in homeostatic phosphate balance. If you then inhibit XPR1 genetically, or in data that I didn't show, with a peptide inhibitor of XPR1, the cell collapses because you have unopposed phosphate import, leading to intracellular accumulation of phosphate and subsequent cell death. So whether we're interested in pursuing this as a therapeutic strategy for ovarian cancer, we'll see whether that works in the future or not, but it illustrates how one can use these unbiased functional genomic screens to identify therapeutic strategies, in addition to the more observational cancer genome sequencing types of approaches for target identification. Now, I didn't go into any of the details of these genome-wide CRISPR screens. It's not that they're trivial to do, but knocking out each of the 20,000 genes in the genome doesn't require doing 20,000 experiments, because it's possible to make a pool of 20,000 lentiviruses, each targeting a different gene, and then infect cancer cell line with that pool of guide RNA, CRISPR-Cas9 guide RNA library. So you're really doing one experiment and using the power of these pooled libraries to get you to the scale that you really need for these unbiased approaches. There's not really a way to do that with drugs or small molecules. There's not a method that I'm aware of that allows you to pool 20,000 drugs together and then do one experiment to test the drug response of a given cell model to all 20,000 drugs. So is there a way around that? Is there a way to get to scale through some clever pooling strategy? And that was sort of the motivation for a clever strategy developed by Channing Yu, a physician scientist postdoc previously in the lab, who said, well, maybe it's not possible to pool drugs, but maybe we can pool cell lines. And we could do that if we molecularly barcoded each of the cell lines just with a random, unique 24 nucleotide barcode sequence that would allow us to then pool many cancer cell lines together and then simply measure the abundance of the barcodes before and after drug treatment. So that if the barcode abundance goes down, and you can measure these abundances either by next generation sequencing or by hybridization type methods, details don't matter. If you see the barcode abundance go down, you can infer that that cell line must have been killed. Whereas if a barcode is maintained following treatment, you can infer that that cell line must have been resistant to a given drug. So we call this method PRISM, and we've now barcoded about 1,000 human cancer cell lines in this fashion. So now you've got this library of barcoded cell lines that you can pool. What are you going to treat them with? So here, Stephen Corsello, a medical oncologist, postdoc in the lab, who's in about 2 weeks going to start his own lab at Stanford, said, well, why don't we just treat with all known drugs, FDA-approved drugs and drugs in clinical trials? And he quickly learned that actually there's no place you can go to actually establish such a collection of drugs. And so he had to go to more than 75 different chemical vendors to establish a library of small molecule compounds, mostly FDA-approved drugs, some that were approved by other like European agencies, some currently in clinical development, so that we have a library now of about 6,000 such drugs available for systematic testing. And we call this part of the Broad's drug repurposing hub. So then the combination of this library of drugs plus the PRISM barcoding method could allow Stephen and colleagues in what's now a PRISM platform to do the following experiment. Let's take the first 4,500 of these known drugs and treat 578 cell lines with each of those 4,500 drugs and ask, do any of those drugs kill any of the cancer cell lines? That's the experiment enabled by this PRISM method. And I'm not going to go to the details here except to say, yeah, kind of as you expect, known chemotherapeutic agents kill some of the cancer cells. Known targeted cancer agents such as, Gleevec and EGFR inhibitors, RAF inhibitors, and things like that. Yeah, they killed some of the cancer cell lines as well. What was perhaps most interesting is that there was a significant number of non-oncology drugs, that is, drugs that maybe have been FDA approved but not developed for oncology indications, showed some activity in this initial primary screening assay, which admittedly was done at a relatively high concentration of 2.5 micromolar as a primary screen. So how do we look at this data set of 4,500 compounds in 578 cell lines? Well, one now common two-dimensional reduced dimensionality representation of this data is a UMAP projection, which many of you have probably seen most commonly from single-cell RNA sequencing data. You can use t-SNE or other PCA type projections and give you a roughly similar result. But what's interesting here is now looking at each of these 4,500 compounds, each represented by a dot, and clustering them not based on their similarity in chemical structure, but based on their pattern of killing across these 578 cell lines. And what you can clearly see is that drugs with similar mechanism of action cluster together. For example, EGFR inhibitors in orange in the bottom here, all cluster together, structurally distinct, but having a similar pattern of which cancer cell lines are killed and which are not. I'll draw your attention, for example, to this yellow cluster of glucocorticoid receptor agonists. Not because I think, oh, this is a previously unrecognized, great cancer drug. No, it's not actually, because the extent of growth inhibition by these glucocorticoids is pretty modest. But you can see this coherent biological signal by virtue of their coordinated activity across such a large panel of cell lines. So even a weak signal that you would never make much of if you were just looking at a cell line or two cell lines or three cell lines, you look across hundreds of cell lines and you can see a consistent pattern of viability defects. And I don't have time to discuss it today, but researchers are increasingly using this data set, the entirety of which has been made publicly available, not so much for finding new cancer therapeutic targets, but as a way to understand protein function based on subtle patterns of viability defects across this large data set. Now, I want to give one example of a discovery that we're pursuing, still working out in particular, and that relates to multi-drug resistance. A protein called MDR1 also goes by the official gene symbol ABCB1. MDR1-based drug resistance has been known for 20 years probably in oncology. It's a common mechanism of resistance for many drugs, for example, paclitaxel, Taxol shown here. And so in this heat map representation, we're seeing the expression of MDR1 across the panel of 578 cell lines, each vertical bar being one of a difference of cell lines. And below that, we're showing a heat map, not of expression, but of drug sensitivity across those same 578 cell lines, where these drugs kill, shown in blue, or they do not kill, shown in white. And you can see very clearly that where MDR1 is high, you see lack of killing or resistance to, for example, Taxol. Whereas if MDR1 is low, you see potent killing. And this is, of course, what you'd expect based on the known mechanism of resistance engendered by MDR1. That makes you feel good about the PRISM system, but it's essentially rediscovering something that's been known for 20 years. What's a bit more interesting is a single drug in the collection that had the opposite pattern. That is, it only killed cell lines that are MDR1 high, and it spared cells that were MDR1 low. Turns out this compound topoxylin with a trade name Zubrin has been FDA approved for dogs for osteoarthritis. It was developed as a cyclooxygenase, a dual cyclooxygenase and 5-lipoxygenase inhibitor, but we're able to show both through chemistry experiments, with SAR studies, and looking at other cyclooxygenase and 5-lipoxygenase inhibitors that this pattern of killing has nothing whatsoever to do with this intended activity of cyclooxygenase or 5-lipoxygenase inhibition. That is, the cancer killing activity is an off-target, unintended off-target activity. In this case, something that we might try to exploit in the future rather than something to be avoided. But what's the connection to MDR1? Is that just some like anonymous biomarker that was picked out of these predictive models, or is there actually some functional connection? And so to address this, Stephen and his colleagues turned again to genome-wide CRISPR modifier screens, in this case, treating cells with topoxylin, the drug, and then doing a CRISPR-wide knockout, a genome-wide CRISPR knockout screen to ask, are there any genes in the genome which, if knocked out, will rescue the cells from killing by this drug topoxylin? And the result was very definitive here. You don't need a machine learning model to see here that ABCB1, that is MDR1, is the outlier in this case of that is, you knock out ABCB1 or MDR1, you're able to confer resistance to this topoxylin drug, indicating that ABCB1 or MDR1 is not just some anonymous biomarker, but is mechanistically key to this mechanism of killing. And we're working that out at a biochemical level, still a bit mysterious. Now that particular hit is obvious. I think where there is going to be enormous value in using more advanced computational approaches is understanding, for example, what story is being told by these less strong hits, where I'm convinced that the biology is going to wind up also being relevant and instructive, but we need better ways to recognize what biological story is being told based on the identity of those weaker hits. So we're going to try to figure out whether is there a path to turn topoxylin into a better drug that retains this anti-cancer activity and actually engineers out the lipoxygenase, cyclooxygenase activity that was for which the drug was initially developed. Now, I wanted to also say that this PRISM idea of pooling cancer cell lines can be extended in vivo. And I was particularly interested in this because there's often a bit of a trade-off that I kind of alluded to this in the clinical data versus experimental data, but there's often a trade-off between, am I going to maximize on physiological relevance of a model system or maximize on the high throughput nature of the big data that one can generate from really tractable, but perhaps less physiologically relevant model systems. And so the experiment of Xin Jin as a postdoc in the lab was to say, well, could we take this pooled cell line model PRISM idea and extend it into the in vivo xenograft setting to understand, not just with your favorite 1 or 2 or 3, your favorite cancer models, but at the scale of like 500 cancer cell lines, could we understand the ability of particular cancer cell lines to survive in different tumor and microenvironments, that is, to survive in the brain or the lung or the liver or the kidney and so on. So that experiment was pretty straightforward. Take this pool of 500 barcoded cell lines, inject them into the left ventricle of a recipient immunodeficient mouse so that the cell lines directly entered the arterial circulation. And then a couple weeks later, harvest these organs, extract DNA, and simply count barcodes, which if you see a barcode representing that cell line, you can infer that, oh, that particular cell line must have been able to survive in that organ. And so this then develops what we're calling a metastasis map or a MET map. And you here shown in this spider plot representation, we kind of in one fell swoop generated a map of which cell lines are able to metastasize to which organs. Here, just a subset of the map focusing on breast cancer. You can see that some cell lines, for example, in this example, HCC 1143 doesn't metastasize anywhere, whereas in the upper left, MDAMB 231 is an example of a cell line that can metastasize widely, whereas other cell lines, for example, HCC 70, that goes selectively to liver, others selectively to brain, and so on. And this is also all publicly available data, underlying data, and a visualizer available at the DepMap website. So then you can start to ask interesting questions like, let's take breast cancer cell lines, breast cancer cell lines, some of which are able to metastasize to brain, and some of which are not. So if we compare those, the genomic features of those cell lines able to metastasize to brain compared to those that are unable to metastasize to brain, and we have the benefit of all these different data types, genome-wide mutation data, copy number, metabolite, RNA-seq, and so on. I will summarize this by saying that there was a very coherent story that emerged, which was that there was a distinct pattern of lipid metabolism in those cell lines that were able to metastasize to brain compared to those that were unable to do so. How did we know that this was a lipid metabolism story? I think the honest answer is that it's because we recognize the names of many of the genes and metabolites involved in lipid metabolism. That's good in a way because, a nice story came out of this, but it's obviously very limited. And so I think a next, a real power of what we should be thinking about advanced computational approaches for is, okay, maybe this is the low-hanging fruit of this data set, but there are going to be many, many more stories behind this, including pathways and processes that don't yet have labels on them that make them readily recognizable. And we need to learn those biological relationships from the data. And I think that's where modern machine learning methods, when applied to data sets like this, are going to be really exciting. I'm going to skip this these follow-up experiments and tell one last story, again relating to drug resistance, because in the clinic, an all too common scenario is that patients may have an initial response to drug treatment, but then the tumor recurs in a fully drug-resistant state. With respect to targeted therapies, some of that drug resistance, maybe a lot, is explained by secondary mutations, often in the target of the drug itself. For example, that's the case with imatinib or Gleevec in chronic myeloid leukemia. Well, that doesn't explain all drug resistance. And what's really interesting is that there are non-genetic mechanisms of resistance that are being discovered. And one that was particularly interesting, and this is the work of other groups, not mine, I'm just showing here to illustrate the breadth of the observations, is that a switch to mitochondrial metabolism, often oxidative phosphorylation-based metabolism, is observed in cancer resistance across multiple classes of anti-cancer agents, some of which are listed here. And so the interesting question is then, okay, well, that's bad, that cancer cells are able to switch into this mitochondrial metabolism state and make some drug resistance. But the question is, in making that switch, do they actually acquire a new vulnerability, a new dependency that we might be able to discover and exploit? And so that's been the work of Peter Tsvetkov, who joined as a postdoc and now stayed on as a staff scientist. He did two types of drug screens in parallel. I'm not going to explain them in great detail, but they get at this drug-resistant, high mitochondrial state phenomenon, and ask, are there any drugs in these drug screens that are selectively kill cancers when they're in this high mitochondrial state? And the answer is yes. And what was clearly at the top of both of these screens, two distinct, very different types of screens, were drugs known as copper ionophores, exemplified by a compound called alesclamol, structure shown here. So these are copper binding compounds. We then ask, well, how do these cells die? Is it by the usual mechanisms of cell death? Short answer, no. We tested pharmacologically, genetically, all the known inhibitors of, for example, apoptosis or necroptosis or ferroptosis, an iron-dependent mechanism of cell death. And none of those could rescue killing by alesclamol. So we think this is a new form of cancer cell death. We're calling it cuproptosis because it's copper-induced cell death. Journal editors hate that idea and they're not letting us use it in the paper, but nevertheless, that is what it seems to be, a unique copper-dependent cell death mechanism. But what does that have to do with mitochondrial metabolism? I had no idea. So again, turned to these unbiased genome-wide CRISPR modifier screens and asked, okay, let's treat cells with this two different drugs, lescomol and another structurally distinct other copper ionophore, and ask, looking across the entire genome, which genes, if knocked out, can rescue from these copper ionophores? And we got exactly 7 genes out of that screen, shown in blue, that can rescue killing. And all 7 are part of a pathway that I'd never heard of, called protein lipolation. That is a lipid modification of what turns out to only be about 6 proteins in the entire genome, all in the TCA cycle, involved in oxidative metabolism. So it's a lipid modification that's required for enzymatic activity of the TCA cycle. What does that have to do with copper, though? And what Peter was able to show is that these lipoylated TCA proteins, if they're highly expressed, we can show in a copper column, but not other metals such as cobalt or nickel, that copper binds directly to these lipoylated proteins, establishing the connection between mitochondrial metabolism and these lipoylated proteins in the TCA cycle and copper. And in support of that, Peter was able to show that if you strip out all of the metals from the cell culture media and then add them back sequentially, only the addition of copper allows this compound lescomol to kill cells at nanomolar concentrations. Add back other metals, iron, nickel, zinc, and so on, and the cells are perfectly happy. It also turns out that there's a really good antibody for these lipoylated or lipoic acid modified proteins, and so you can use them to stain tumors by immunohistochemistry. So you can now have a biomarker of which tumors are lipoylated protein high, reflecting the high mitochondrial state, and which ones are low, with the therapeutic hypothesis being that we should be treating tumors or patients with this class of drugs with when they're in this high mitochondrial, high lipoylated protein state only, and not treat those tumors that are lipoic acid low. And I mention this because there's a quite famous story that's an interesting read published in the New Yorker, written by Malcolm Gladwell in 2010, that was the story of the development of lescomol as a drug. Actually went into the clinic in cancer, initially showed some inklings of promise, but there were no biomarkers of who is the right patient population to treat. So something kind of random was treated that we now think has no basis to believe that there's a connection to this biology of high mitochondrial lipoylated protein state. And so the drug failed in phase 3 clinical trials. And so we're interested in thinking about, can we optimize a lescomol or other copper ionophores, not just to treat in randomly selected cancer patients, but rather patients that have a biomarker of this specific state. And that's really, I think, the concept of cancer precision medicine with novel agents that we want to get to. So I won't read all of these conclusions, except to say we're trying to push around the edges of how can we make high throughput biology, genomics, functional genomics, chemical biology. How can we bring this to bear on increasingly physiologically and clinically relevant model systems to both discover predictive models of which patients will respond to which treatments, and to actually discover novel therapeutics and novel targets for future drug discovery. I want to thank the people that have done all this work. PRISM project led by Stephen Corsello, the METMAP by Xin Jin, this last cuproptosis story by Peter Tsvetkov, I started with the story about non-canonical proteins, the work of John Prenzner, Dan Bondison on the XPR1 ovarian cancer story, and with the permission of Nick Wogley, I shared his work on Count Me In, the direct-to-patient story. With that, I will stop. Thank you very much. Happy to answer any questions if there are any.
Outstanding talk, Todd. Thank you. And indeed, we do have a few questions. I have some of my own, but I'm going to jump in with some that are in the chat just to get us started. So one from Felix Dietlein. Amazing talk, Todd. So inspiring. Currently, our assays evaluate the efficacy of a drug that are largely based on proliferation. I was wondering whether other functions, for example, pathways in metastasis, unrelated to proliferation, could also be explored in a scalable way.
Yeah, so that's a very insightful question. In particular, that last sort of question about being scalable. So viability is a highly scalable readout. It happens to be not a bad, phenotype for cancer cells, where killing cancer cells is, not a bad idea. However, there are probably a lot of other approaches that one could imagine. For example, could you use single cell RNA sequencing as a phenotype to have a non-viability but potentially scalable readout of perturbation? There probably are other things. I think there's also interest in extending these kinds of genome-wide CRISPR screens into the in vivo setting, where it's kind of still a proliferation assay, viability assay, but in the context of a more physiological microenvironment. So you're also testing the dependency of a cancer cell model on the local microenvironment. So it's still related, but yeah, I think there's a lot that could be done in that's not been done yet in that direction.
Could you talk a little about what the decision-making process would be for you and your colleagues at the Broad? What's the threshold over which you would invest in, in actually doing those measurements in a massively scalable way? How do you pick your winners?
Yeah, it's an interesting question. Usually, it's a combination of, pilot data that suggests that, this is a viable strategy, and a champion that really wants to drive something to scale. My experience has been that if there's a great idea, some preliminary data to support it, and a champion and a truly scalable method, which many things aren't, there's a way to find the resources, and if you have to create the capabilities to create that data set, that's possible to do. But you need those ingredients first. Do you always, are data sets always as informative as you hope? No. The challenge, of course, is that you can't know that until you create them. That can get you into a dangerous loop of, we don't need to think about this, whether this is useful, just big data is always good. It's not always good. And so I don't know that there's a magical solution to that. I think increasingly trying to think about what are data types that would really be orthogonal to what we're learning from existing things like viability assays, I think are going to be useful.
Great. So here's another question, somewhat philosophical or sort of high level. Regarding the scalability problem of precision medicine, how do you see real world data use cases as being a solution to address this issue?
I think that's part of the sort of thinking behind the Count Me In initiative, is to try to exactly use real world data. I think honestly, the future of clinical trials could look very different than it does right now if we figured out how to use real world data more effectively. And there are people I'm sure on this Zoom that are much more knowledgeable about this than I am, but I think could be really powerful. Does it get you to the new approach as opposed to using real world data to understand which of existing approaches that are out there being deployed in the clinic are most effective? That I'm not sure. So will it be a great hypothesis generator for the next great target or strategy? Maybe, but I think that's less certain.
Here's another interesting philosophical question. How important do you think it is to understand a disease to cure it? So scurvy was cured by the observation before people knew about vitamin C. How much should we invest into understanding cancer versus observational studies to advance treatment?
Yeah, that's a really interesting question, and one I think a lot about. So I have what maybe you may find an unsatisfactory answer. I don't think that it's necessary to have a complete encyclopedic understanding of cancer biology, or any disease for that matter, in order to effectively treat it. However, I don't think that we'll make progress without at least a scaffold of understanding of what makes, in this case, cancer cells survive. And so I think it's somewhere in between. I kind of believe that we should strive to learn the minimum possible about cancer biology, for example, to be able to develop effective therapies and not feel like the entire textbook needs to be written first in order to advance therapies. But I'm not optimistic that if you tried to do this entirely empirically, that much progress would be made.
Let me ask another question. As these different assays, different therapeutic approaches emerge, do you have a vision? And I'm asking you this because the Broad does do scale well, but at how in the healthcare system we can get these tests and treatments paid for when they're extremely individualized. Do you interface to that part of the world?
Yeah, so I mean, I'm not close to the reimbursement side of things. However, the $100 whole genome sequence is coming. And so I think with that being a reality, I think the whole notion of these highly specialized genetic tests is just going to go away. And it'll be a test, and it'll cost, hundreds of dollars, not thousands of dollars. And at $100, let's forget cancer for a second, but just, common disease, for example. At hundreds of dollars, it's going to make sense for, every individual, maybe every newborn, to have their genome sequenced, to have that part of the medical record. And I think that will stimulate a crop of new companies that are building tools, interpretive tools, and on top of those data. So I don't think that the cost of genetic diagnosis is going to become a barrier. I think the greatest barrier is going to be lack of knowledge and understanding of what the genetic sequence means. And for that to happen, we need a path for large-scale whole genome sequencing to occur and for the data to be shared. So a scenario that I think would not be good is if individual companies, hospitals, whatever entities start engaging in this and then keep the data siloed, particularly in an effort to monetize it or sell it to pharmaceutical companies or what have you. And we never get to the scale that's really needed to make the connections between those genotypes and, let's say, the entire electronic health record. So I think it's good that companies are entering this space, but I'd like to see a path whereby they can build their tools on top, and that's where the value is, but they're monetizing those tools, not monetizing the underlying data, and that patients should be able to say, hey, I wish to contribute my data, whether it's EHR data or genomic data, or hopefully both, to qualified researchers, and to have a mechanism for those data to be shared to support discovery. Because, you know, generating the sequence affordably is going to be the easy part. Interpreting it is tougher.
Absolutely. And how about within health systems? Do you see enough data being shared from the genomic sequencing that is emerging from research and clinical endeavors?
I assume that's a rhetorical question. No, I think it's not being shared enough. I think it's not being generated enough, and too often. I mean, I think things are moving in the right direction of some sharing, but you know, in the end, these data belong to patients, not to hospitals or payers or anyone else, and that's why I think this whole idea of making it possible for patients to say, hey, I want to share my data, and have a button they can press to make that possible. I know, you know, you and others have been thinking about this for a long time. That should be the standard, you know, across the country. And it just feels like we're not close to that in most health systems.
All right. Well, that's a good agenda to continue to pursue. And I'm really pleased to see your full support of that approach, because I do think we need the data. And I do think the data versus the tools are completely separate buckets. That's fantastic. So Todd, thank you very much for a really comprehensive look at work going on across the road at characterizing cancers. And thank you to our participants in the series today. I'll just give a quick preview. We've got Rob Knight, co-founder of the American Gut Project, and Vinay Prasad, associate professor at UCSF, coming up, and also Chrissy Farr, formerly of CNBC, now a health tech investor. You can see more about these upcoming talks at chip.org events. We have a robust program and set of faculty who are interested to interact and learn together. So feel free to keep in touch with us, and we will see you all at the next Landmark Ideas talk. Thank you very much. Thank you, Todd.
See you. Bye.